Hello, USMLE candidates. Welcome to another video. Today we're going to be taking a look at genetics. And why we are discussing genetics today? Well, I think this is one of the more obscure subjects and parts of your USMLE Step 1. And I think a little bit of discussion on it or a series of discussion on it would help us quite a bit in this scenario. So I'm just going to solve a question in front of you, maybe discuss some high yield fact, and tell you what you need to know to get genetics right on your test day. So let's move on. And I'll also be discussing where you can study this topic from the book as well. So that's going to be a bit later on in the video. So which of the following feature is characteristic of that strand? A 4-year-old boy is brought to the clinic because of increasing fatigue and diffuse bone pain. A physical examination reveals generalized lymphadenopathy and multiple purpuric lesions over the extremities. Laboratory studies demonstrate anemia and thrombocytopenia in peripheral blood smear analysis identifies lymphoblasts. Further evaluation confirms the diagnosis of an acute lymphoblastic leukemia, and treatment is initiated with a chemotherapy regimen containing doxorubicin. The medication intercalates into DNA and disrupts DNA replication, a process that occurs at replication forks and generates two newly synthesized daughter strands. One of these strands is produced in the direction opposite to the advancing replication fork. So the following features is characteristic of that strand. So this is the classic genetics question, and this per se is not difficult, but it does look to be difficult when we are solving UWORLD for the first time, because many of us are in our fourth year or final year or third years, and we can't really remember the basic concept that we studied in DNA transcription or microbiology or molecular biology or in biochemistry. So just need to understand what we need to know. So in these questions, I generally have an approach to just find out the fact that the question wants us to know. So what does the question want us to know? The question wants us to know that just the following features is characteristics of that strand. What strand are they talking about? So one of the strand is produced in the direction opposite to the advancing replication fork. So you need to understand what they're talking about. So this is their concentration, right? The main question starts from this line in itself. All of this is not important. This is just your context that they've given you, and this is not important. So you're not supposed to know what doxorubicin does. You're not supposed to know that we use it in acute lymphoblastic leukemia. You are supposed to know that if you're sitting for step 1. But for this question, you're not supposed to know that. They're not testing that. What they're testing is that they're telling you, they're asking that whether you can understand what is being asked in the question, and then whether you know about that process yourself. So it is good to know what doxorubicin does. They have already told you that it integrates into the DNA and disrupts DNA replication. It is good to know that if you're sitting in the exam, but what is more important to know is what they're actually talking about. So what happens is that there are two strands in the DNA replication process. So let me show you. So there are two strands in the DNA replication process. Uh, one strand is this. So this is the original DNA. The DNA has two strands, and both of them are anti-parallel. So both of them start in a different direction. One of them starts from 5 prime to 3 prime, and the other one starts from 3 prime to 5 prime. So this is known as anti-parallel. Anti-parallel means that they're going in opposite direction. They are parallel, but they're going in opposite direction. So if they were going in the same direction, that's if this was going from 5 prime to 3 prime, this was also going from 5 prime to 3 prime, then we would call these strands parallel, but they're going in different directions. So we call them anti-parallel. Now, this much they have told you in the question, right? This much that they've already told you in the question is that uh, DNA replication occurs in the replication fork and generates two new synthesized daughter strands. This much they have already told you in the question. Right now they're asking that which of the following feature is the characteristic of that strand that they're mentioning is the one produced in direction opposite to the advancing replication fork. So you need to know what this is, this part. So this is a replication fork, right? This divergence, this V-shaped divergence over here at this point is the replication fork. So this one, at which the DNA is separating, this is the replication fork. Now, there is one strand that is opposite to the replication fork, and there is one strand that is going in the direction of the replication fork. So if this is the replication fork, and this DNA is going to continue to grow this way, if you can see my cursor, the DNA is going to, the replication fork is continuing to go this way. So it's going to continue this way, right? So there is a formation of a strand that is forming in the path of the replication fork. So obviously, whatever strand we're talking about must form in this direction, the direction in which my cursor is going. And there is only one strand that is forming in this direction. This strand, you can see the arrow over here, right? Even if in this picture, this is the replication fork. So we need to identify which strand is forming in the direction of the replication fork. And you can see this strand is forming in the direction of the replication fork, right? This is going away from it. This is going towards it. Now, whichever strand goes towards the replication fork, that strand is known as the leading strand. And whichever strand is going away from the replication fork, that strand is known as the lagging strand. Now, what is the concept behind naming, behind such naming of the strands is not important, but this is what they want you to know is that the strand going towards the uh, triple application fork is the leading and so going. So I hope you know what this strand means. What happens is that, of course, there is a formation of new DNA. So now what happens is that, let's say, for example, this is the old DNA, right? This is the present. Now we want to form a new DNA from this old DNA. So what we do is we first separate it, and during the separation, there's an enzyme which is known as DNA polymerase. And DNA polymerase, what it does is it takes one strand and forms its copy. So it forms another strand that is anti-parallel to it. So this strand was starting from 3 prime. DNA polymerase, which is in the blue, is forming another strand. And likewise, in this direction, is also forming another strand. So from one DNA molecule, we get another DNA molecule, right? This is how DNA basically replicates. This is the DNA replication process. You can understand it better from other videos, but just for the sake of completion and making it concise, this is the process of DNA synthesis. So now what they're asking is, okay, uh, one of these strands is produced in the direction opposite to the advancing replication fork, right? So one is towards the replication fork, and the other is in the uh, direction opposite to it. So this strand is in the direction, this strand is in the direction towards it, and this is in the direction opposite to it. Okay. Now, one thing that you have to know is that DNA polymerase forms DNA in the direction of 5 prime to 3 prime. That's its function, right? It's like its function. So it forms DNA from 5 prime to 3 prime, right? If we look at first state as well. So DNA polymerase 3, which is the DNA polymerase in our cells, forms a 5 prime to 3 prime synthesis. Okay. So it cannot form a DNA strand from 3 prime to 5 prime. So obviously, this strand, the leading strand, has no problem because it is forming in the direction of 5 prime to 3 prime. But the lagging strand has a problem because it is forming in the direction of 3 prime to 5 prime. But since the DNA polymerase cannot form it, so what it does, it starts forming it from 5 prime to 3 prime. But the problem is that the strand cannot be increased from this side, it has to be increased from this side. So what it does is that it forms the lagging strand in parts. You can see it forms it in parts. You can see the red one, it forms it in parts, right? Because what happens is you have to use a bit of your imagination now. Because if it forms the strand in parts, let's say it was attached, let's say, let's concentrate on my cursor, let's say it was attached to this part and it formed a strand in here. Now it needs to get detached and go towards here and then form another strand, gets detached and go towards here and forms another strand. So it keeps on doing that until it copies the whole strand in this specific pathway. But what it does is this: it creates these fragments, and these fragments are known as Okazaki fragments, right? And these fragments are formed, which are later joined by DNA ligase, uh, which is this enzyme. So you can view this page also. This is in your first aid. This is page number 36 in the biochemistry section. So this forms phosphodiesterase bond between Okazaki fragments. Okay, so that's what they're basically asking. Now, give you the question: which of the following characteristic, one of these strands is produced in a direction opposite to the advancing replication fork? Which of the following features is characteristic of that strand? And you know already that they're uh, talking about the lagging strand, and this is the correct answer then. Production of multiple short DNA segments. Talking about the Okazaki fragments. Also, I have the explanation right over here. You can read it, but it just contains all the things that we have discussed. So you can pause the video, you can read it. You can also read the incorrect options. It's written over here. So education objective is DNA synthesis occurs in the 5 prime to 3 prime direction only. During DNA replication, one daughter strand is synthesized continuously towards the replication fork, whereas the other daughter strand is synthesized discontinuously away from the replication fork. The lagging strand is formed from short stretches of newly synthesized DNA separated by RNA primers, known as the Okazaki fragments. So this was it. This was a short concept to know what this question is asking.